Good morning, good afternoon, everybody. Welcome in from across the continent today. My name is Jesse. I'm here with Exploring by the Seat of Your Pants. And I think we have all familiar faces in the crowd today, which is fantastic. But if you are joining us for the first time, we're all about bringing conservation, adventure, and science into classrooms around the world. Now, this program today marks the second part of a special 10-part series we've got in conjunction with the amazing folks at the Gairdner Awards. The Gairdner Foundation awards the coolest scientists and explorers in neuroscience and biomedical and health research around the globe. Many of their laureates have gone on to win the Nobel Prize. We've had the chance to feature their amazing stories for years, and I can't encourage you to check out Gairdner.org and at Gairdner Awards on social media enough. Lots to explore and discover there. Now, last year, we had the chance to bring on Dr. Ed Boyden, and he is... You know what? I could list a whole bunch of accolades and awards he's won over his early career, but I'll just say this. I've had the chance to host over 1,500 broadcasts, and Ed's presentation last year was one of the only talks we've ever had in the history of Exploring by the Seat of Your Pants that absolutely left me gobsmacked. So you're in for a real treat hearing about some really, really cool work with his Synthetic Neurobiology Group at MIT. We're going to learn a little bit about what he does to understand and heal the brain. So without further ado, Ed, thank you so much for joining us again, and welcome back to the broadcast.
It's great to be here. Yeah, thank you so much. So I'm here to talk about our brains. So, of course, they sit in our heads, and they mediate our thoughts and our feelings, and they make us who we are. But what is the brain, really? Well, it actually turns out to be an incredibly complex machine, maybe the most complex thing that humans are currently thinking about exploring and studying. And the reason is twofold. One is the spatial complexity. So brains are made of cells, and the cells are enormous. Most of us think of cells as tiny little things, right? Blood cells wandering our body. Brain cells are huge. Some of them are a meter in length, going down our spinal cord. The ones in our heads go centimeters in each direction, much bigger than a blood cell. But brain cells have weird shapes, like trees. They form branches. And the branches are nanoscale. We just zoomed in into one tiny piece of one branch. And a branch is full of molecules, themselves nanoscale. The human genome, which is our blueprint, contains recipes for 30,000 genes. And each of those is a tiny biomolecule, right? The genes encode tiny biomolecules, nanoscale objects, right? A billionth of a meter in length. So how do you understand something big like a brain in terms of its tiny, tiny pieces, which come in thousands and thousands and thousands of different flavors? But it's worse. Time. Brain cells are very high-speed biological machines. So if you form a memory and recall it 5 years later, if somebody gets old or develops Alzheimer's disease, those are slow processes. But brain cells fire off little electrical pulses, and they last only 1,000th of a second. So sometimes I half-jokingly say we only have to solve two things to understand the brain: space and time. Now, why? Well, the brain is what makes us who we are, right? It encodes our feelings and our thoughts and our decisions. But there's another reason: brain diseases, Alzheimer's disease, stroke, ALS, epilepsy, spinal cord injury. The list goes on and on. No brain disorder can be cured. And the treatments aren't that great. A lot of them are hard to treat, and the treatments only work for some people, and the treatments have side effects. Over 1 billion people around the world suffer from something related to the brain. Not surprisingly, because the brain is complicated, it's hard to treat brain diseases. A brain drug will take 10 years to go from the lab to the market. The failure rate to get clinical approval is almost 100% failure. The cost is billions and billions and billions in cost. And even if you get through those hurdles, there's a good chance the drug doesn't work very well. So today, I want to tell you two short stories, one about space and one about time. And the idea is that we can build technology to understand the brain. Let's start with space. As I told you, brain cells are gigantic objects, but the wiring of the brain, the molecules of the brain, are tiny. They're nanoscale. How can we understand such a thing? Brain scans are very popular. You might have seen images like this in the news. They're monitoring blood flow in the brain. On the one hand, it's non-invasive, but at the other hand, it's blood flow. It's not the actual computations of the brain. How could we possibly see something as small as a brain cell? Well, in our group, we often try to be the opposite of others. And so one of our ideas was take a brain, fill it with baby diaper material, the same kind of swellable stuff you find in baby diapers. Add water, and we can make the brain as big as you want, 1,000 times bigger. So in panel B, that's a small piece of the mouse brain. Panel C, a day and a half later, we blew it up 1,000 times in volume. Here's a movie of a piece of the brain being expanded 100 times in volume, actually. So we formed the baby diaper polymer inside the brain earlier through a chemical process that I'll tell you about in a second. And then we add water, and I hope you'll see that we add the water right there. This is a time-lapse movie, half an hour sped up to half a minute. But I hope you can see this piece of the mouse brain is growing before your very eyes. Okay, that's what it looks like from the outside. What's happening deep, deep inside? Well, this cartoon illustrates what we are going for. We want to take a brain cell like this golden one on the left and make it look like the thing on the right, a constellation of biomolecules hovering in space. The relative organization has been preserved, but they're all pulled apart. If two biomolecules were touching, they're now separated, and we scaled up by a linear factor. To make this work, we had to invent several chemistries, shown in the following cartoons. Here in brown are proteins. We invented handles, shown in purple, to bind to the proteins. The handles let us pull the proteins apart. Next, we have to add the baby diaper polymer. Now, to get it inside the brain is hard. So we add monomers. Those are the little white spheres. They self-assemble into the polymers, the long white chains, through a chemical process called polymerization. And when the long white chain encounters a handle, it forms a bond. Now, if we add water, the baby diaper material would swell, and because of those handles, the molecules would get pulled apart, right? Well, not so fast. There's one more step. We have to soften it. We can add detergent or heat or enzymes to chop stuff up. Then, when we add water, the baby diaper material swells, but because of the softening, because of the handles, and because of that polymer, we can pull all the biomolecules apart. Here's a piece of the mouse brain. A lot of neuroscientists study mice. We've given every brain cell in this image that you can see a fluorescent protein. That is, a glowing protein from jellyfish or corals. That way we can color code the brain cells and we can tell them apart. So these different colors are because different cells are expressing different fluorescent proteins from corals and jellyfish. I'll play the movie. We're going to fly through the brain. This is a part of the brain involved with encoding memories, amongst other things. You can imagine reading out what a memory really is by looking at the pattern of wiring of the brain. We expanded this 4 times in each direction, 100 times in volume. I hope you can see the wires are distinct. You can see them because they're color-coded and expanded. Here's a fruit fly brain, 100,000 neurons. We're going to fly through it now. You can see how we can image across scales. We can see the whole brain, but now we can also see individual wires. My dream is we can make maps of the brain so detailed we can simulate them in a computer. Imagine a computer simulating actual thoughts, feelings, and decisions. That's the goal of our group going forward. Now, that's our attempt to map the spatial dimension of the brain. But what about time? The diaper expansion method I told you about will not work on a living thing. We have to preserve the brain to expand it, so we cannot get any time information. The brain is such a high-speed machine. Well, we can use light to see the brain and control the brain's electrical activity. Here you can see brain cells blinking at you. This is a cartoon showing the electrical pulses made by the brain. What if we could put solar panels into brain cells? Then shining light would allow us to control the electricity, right? Light hits the solar panel, electricity results. Electricity is what controls the neuron. Then we can bring light into the brain. Electrodes are brought into the brain, even of awake humans during surgery for deep brain stimulation for Parkinson's disease. Why not an optical fiber? The tricky part, and this is what I shared the Gairdner Award for, is to make brain cells respond to light. How? Well, it turns out organisms all over the world respond to light and convert light to electricity. This is a single-cell green algae we're looking at. It swims in bodies of water. We just zoomed into its eye spot, a little organelle deep inside the algae. It contains light-activated proteins. When light hits them, charged particles move through them. This is exactly what we want, right? Sort of acts like a solar panel. Now, this is where we got lucky. Turns out that this is a protein, and the protein is encoded by a short gene. So, can we take the gene that encodes for this protein and put it into the brain, let's say, of a mouse? We took the piece of DNA that encodes for this protein and put it into a viral vector, a gene therapy vector. Then we could deliver the gene to a brain cell of a mouse. Amazingly, the mouse neurons could manufacture the protein, even though it was from a plant. Furthermore, it could install the protein properly and have it work in the brain environment. This was sheer luck. It didn't have to happen, right? The protein could have been toxic. Now, when we shine light of the right color, in this case blue light, on the brain, we can activate those cells and drive them to fire electrical pulses, just like in your brain right now as I say these words. Many biologists use, we call it optogenetics, opto for light and genetics because these are genes, to study the brain. By the way, we share all of our tools freely. Thousands of people do expansion microscopy, the method I told you earlier, and thousands of people do optogenetics. I'll give you an example. Dopamine. You may have heard of dopamine. Some people call it the pleasure center of the brain. But it's more complicated than that. It's involved with learning, movement, and lots of stuff. We wondered, could we drive these neurons and see what they do? So using genetic tricks, we can make the dopamine cells convert light to electricity, so they would be activated by light. Now, dopamine, as I mentioned, does get a reputation of being the pleasure center of the brain. We wondered, if you activate these cells with a pulse of light, will you make the brain do more of what it was doing? Here's a box. If the mouse pokes its nose in the right portal, it'll get a pulse of blue light from a laser down a fiber into the brain. If the mouse pokes its nose on the left, nothing will happen. Here's the result. Mouse pokes its nose, gets a pulse of light. Pokes its nose, get a pulse of light. Pokes its nose, get a pulse of light. This mouse is working for light. So this allowed us to prove that activating these dopamine neurons was enough to make the brain do more of what it was doing. Thousands of people use optogenetics, just as with expansion microscopy, to study the brain. What about using it as a therapy? Many people are blind. They've lost the light sensors in their eyes. Here's a blind mouse. It has a mutation that mimics the human condition for blindness called retinitis pigmentosa. It's in a water maze. Mice like to swim, but they love sitting even more. The mouse is trying to swim to find a platform. There it is. Now the mouse can climb out. It took a long time because this mouse is blind. Here's another blind mouse. Mice are smart. Eventually they learn how to find the platform, but they're not using their eyes to do it. I think you get the idea. Now, let's take these mice and give them one gene therapy dose or viral dose of a gene therapy vector containing our light-activated molecule. In other words, can we convert the rest of the eye into a camera? Here's the result. This mouse was blind a few weeks before. We put our light-sensitive protein in the eye, even though it lost its original natural light sensors. And look, mice can find the platform now. Millions of people have forms of blindness where they lose the light sensors in their eyes. If we could put these light sensors into the rest of the eye and convert the spared part of the eye into a camera, we could help people. A couple summers ago, a team in Europe did just that. They took one of our molecules, which we published in 2014, and they put the gene therapy vector with our gene in it into the eye of a person. The vision was restored partly. Not perfect. He couldn't read very well, but he could recognize objects. He could see doors on a hallway. He could see lines of a crosswalk. He wears goggles to convert the visual world into the right color, in this case orange light, because each light-activated molecule has a best color. And now, if I recall, there are about 30 people walking around with one of our molecules in their eyes. All of them, if I recall properly, are having partial restoration of vision. Each of our tools is very powerful. We send them out into the world, and thousands of people use them to study the brain. But my dream is to simulate the brain in a computer. I want to know what a thought or a feeling really is. So our goal now is also to combine these tools, map the brain, control the brain, and so forth, and make computer models. We want to start with small brains like fish and worms, but our goal is to scale to the mouse and eventually the human. We've been adapting our tools to work in the fish. Here we are using the diaper expansion method to expand the fish brain, or the worm. Here we are expanding the worm, the whole thing. Our dream is to combine these tools and be able to make models of how the brain works. In summary, this schematic shows how we plan to do it. The blue part is we'll image the brain while it's in action and perturb it using the optogenetic tools. In the lower left, we'll use expansion microscopy, the diaper expansion method, and make a map of the molecules. I'm sure you've all heard of machine learning and AI. We're going to use AI and machine learning to figure out the model. For example, from the live data shown in blue, we can make these models shown in pink, where we guess how the network works. We can also try to infer what molecules are there. We know the genome, right? We can try to figure out what molecules must be there to create the activity. Finally, well, we can make a map of the molecules using our expansion method and make the model in the lower right shown in purple, going from molecules to ultimate physiology. So to summarize, we build lots of tools to map the brain and control the brain, like expansion microscopy and optogenetics. We send them all over the world. This is a crowded slide, but it acknowledges the people in the group and our alumni who've led this work, as well as collaborators all over the world who helped us with it. And going forward, one of our top priorities is to combine these tools and integrate them so we can make computer simulations of the brain. I'd be happy to take questions.
Thank you so much, Ed. I mean, professional detachment out the window. It's the second time I've had the chance to hear you talk about this, and my jaw's on the floor the whole time. I mean, it's wild research. It's the leaps that you guys have made, and the way of thinking about it to begin with is it is truly remarkable. So a big thank you to you for that, and for our audience, please do keep sharing those questions in the chat. We've got a few to begin, and one of them was, how did you ever come up with using diaper technology for this? Like, how, I don't know. Was this like a personal experience you had that you went, oh, this is a snap decision, let me see if we can try it? Or how did this even come about?
Excuse me, let me take a sip of water.
Yep.
Well, we knew we wanted to move molecules apart because how else can you label them? And so my very first postdoc in the lab, Brian Chow, and I were brainstorming about how to do this. A second line of thought was there was a very famous MIT professor named Toyoshi Tanaka, and he was working on these kinds of polymers in the 1980s. When I was an undergrad at MIT in 1995, I wrote to him wanting to work in his group. And then finally, we had two amazing grad students, Faith Chen and Paul Tilburg, who were very much focus on the question of how to do nanoimaging. And so when we realized how hard it was to do nanoimaging, we decided to give this a try.
So for our audience's sake, because a lot of our students won't be familiar with this and what an actual research lab is like, from the conception of this idea to you actually implementing it, how long a process was that? Is this days, weeks, months? Can you give us a sense of where you're at?
Well, the initial idea is that we should expand the brain. Probably we started brainstorming around 2007, but we didn't know how important the idea was until 2012, which is when the two then students, Faye Chen and Paul Tilburg, running group, and realized just how hard nanoimaging was. And they were the first to really hone in on a practical way of doing it and give it a try. Optogenetics was the same. Carl Dijsselhoff and I thought up the idea to do this in the year 2000 when we were both students at Stanford, but we didn't give it a try till 2004. So sometimes it takes a while for the knowledge to be converted to wisdom.
I love highlighting this, and this is true of people that go to space. It's true of people who do deep sea exploration. I know when you're a student, when you're in junior school like our students are today, the thought is that, you do a project, it's done in a few months, it's done in a few weeks. That's what you submit for your sort of like final exam. And when you actually are in science, it's quite regular for programs and projects to take many years, 4 years, 5 years, decades in some cases. Certainly in space exploration, that's the standard. So what's kind of nice about that is that when you come up with a great idea, you get to create a project on it that you can share with the world, collaborate with all sorts of amazing people, and you keep highlighting all these collaborators of yours, both in that incredible slide and in your answers. And I think that's one of the real bits of magic of science, is that sort of a simple thing can lead to some really great ripple effects down the road. So thank you for that answer. You showed these incredible videos of the brain and diving into it and worming around. How do we get those videos? What's the deal? How are we creating those that imagery for people to sort of navigate and see what it's like inside?
The cartoons were made by a company in Maine called Sputnik Animation. They do a lot of work with public television and scientists. The data, though, like the actual brain data that I showed you, those we rendered in our own group using computer software that our group has been making.
That's very cool. So, and I love that you partner with sort of a group of artists to visualize this. This is something that I remember back when I was in University of Toronto. I was just general biology, nothing quite like what you do, but to visualize the interactions of cells and of organs in the body is just it's magic in a way that you can't really conceive of because it's so small, right? We can sort of picture an arm moving like this in space, but to see how mitosis happens or something is really quite spectacular when you get the chance to check it out. So thank you for that. And we'll have to check out that company in Maine. We may have to bug them for a future broadcast. You highlighted 1 billion people, which is a remarkable amount of people on this planet with some form of brain disorder or challenge. Is there a specific one that has sort of led the charge in terms of how we're trying to figure out how to help or solve it? Is it something like Alzheimer's? Is there anything you can share in this, or is it just generally across the board we're working to solve everything?
Well, there are a couple answers to that question. I mean, I really think of like common intellectual structures. So in the brain, the problem is you just got to see what's there, right? And that's what our technology lets you do. So we share all the tools freely, and thousands of scientists work with our tools trying to solve problems in parallel, right? So I don't think that there's any one specific disease that is being focused on. In fact, that's kind of the point, right? I wanted to solve all these diseases, but where to start? There's thousands of them, right? So my thought was, let's build a tool, and then everybody can solve them using our tool.
You're a pretty humble guy, so I don't know how you're going to try to take this question, but it's really rare for people to leave their technology, their ideas, sort of open source where anyone can use them and collaborate. That is quite unusual. Is that something that is sort of an institutional thing, or just something that you personally were very keen on? Because it'll allow more inquiry and more success to be had much faster, and it's really a testament to you. So I'm just curious how that idea came about to begin with. So leave it for everybody.
I always find it puzzling when people say that, because distributing tools is the purest selfishness, right? I'm not doing this tool building for no purpose, right? I want to solve all these problems. So for me, the way I view it is I build these tools, the people who are going to take the tools to solve problems, that's achieving my vision, right? So I don't, if I were to hold the tools back, what's the point, right? I might as well go retire and lie on the beach.
I mean, I agree. I like the attitude, but it is a question I'm sure you've had 8,000 times over the course of your career because it is unusual. I mean, when we developed some early scale medical technologies, and I know this just from my reading, the general thought for a lot of things back in the day was like, oh, we've developed this new fantastic thing, and let's make sure no one else knows about it so we have sort of the rights to do it, sort of like we do with proprietary technology, proprietary lots of things in the world, intellectual property. And you are the outlier, but it is a fantastic way of thinking about the challenges in the fields at large. So thank you for that, Ed, and great question, guys.
Don't thank me. This is me achieving my goal, right? So, and I don't. If other people don't do that, I don't know. I'm not here to explain other people's failures. I mean, that's
fair enough.
For that.
Oh dear, I do like purest form of selfishness. That's going to stick with me for. That's how I'll subtitle the talk next time, I think. Ed, we had a question about what your favorite part of your job is. Is it doing nitty-gritty things with cells in the lab? Is it sharing your work? Is it collaborating with other people? Is there anything that jumps out that really gets you up in the morning?
It's all great, yeah. I mean, except for like writing tedious progress reports, I could do without that. But all joking aside, I mean, I mean, seeing something for the first time ever is always fun. Yeah. You know, we did the first light-activated neurons, the first expanded brain. You know, that's a real pleasure of it. Working with great people, of course. You know, and it's seeing people solve problems with it. Right? There's so many therapies now in clinical trials, thanks to us. You know, people have applied our tools to, you know, as I mentioned, blindness already, but also it's inspired therapies for Alzheimer's disease that are in clinical trials. People are trying to figure out how to, you know, help people with all sorts of stuff. You know, it's all part of the mission.
This is a bit of a clinical or clinician's question, but I'll see if you can field it just the same. You talked about the fact that you have this remarkable technology. You showed the mouse thing, which is still wild to me, and that 30 people or so have this right now. Why not millions? Why does it take a while to go from something that we've proven to be effective to mass scale rollout? And this is sort of a broad question for how medicine works in general, but if you can speak to it, that would be lovely.
Well, it hasn't been proven, right? It's not been approved by any regulatory body, right? So the way most medicine works, you have a government agency that regulates whether something is safe and effective, and that's their job. So, I mean, the people doing the clinical trials have to prove it, right?
Yeah. So I like this because we're doing a program, another Gairdner program on vaccine development next week. And so when we rolled out the COVID vaccine, before it was mass adopted by the public, hundreds of thousands of people were involved in the test for that to make sure that it was safe and effective before they said, okay, hundreds of thousands of people have taken this. It is safe and effective. Now millions and billions of people can take it. So 30 is a pretty small sample size in the scheme of things, but it's important to sort of gradually build it up so that we don't roll out something that has inadvertent effects that could be very negative for people. So again, it's early, exciting days for this, and it's just an incredible bit of technology. So I'm keen to see what is going to come of it in 5-10 years down the road. And that's a great question, guys. Ed, just a few more questions. Time flies when you're having fun, so we really appreciated having you. One, which I love when we get this one, is what does your day-by-day look like? Do you have a different day every day? Is it like, are you all over the place? Do you have a pretty standard format of what a day at work looks like at MIT?
Well, I try to wake up early and think and write and deal with emails and so forth for a couple hours. And then from 8 o'clock till 6 o'clock every day, you know, it's meetings with group members, sometimes thesis committee meetings, sometimes faculty meetings. Then I go home. So.
Fair enough. I like that early for you is before 8 in the morning. So this is why you've accomplished so much more than I have to do things before 8. That's incredible.
Yeah.
So
I usually try to wake up around 5 o'clock or even earlier.
Wow, good for you. I also, one day in exploring by the seat of your pants history, we're going to find someone who likes progress reports. You are in a long litany of people in all fields that abhor those. So appreciate that answer.
No, then I can hire
them. Ed, before we wrap up, is there any last message you'd like to leave kids with about neuroscience, about the work that you do, anything that they could take to encourage them to learn a little bit more and dive in with the field more broadly?
Oh, I have endless thoughts about that topic. Where to begin? Well, I wrote 2 essays. If you Google them, you can find them. One's called Assumption-Proof Brain Mapping, and one's called Architecting Discovery with Adam Marblestone, used to be in my group. And yeah, I really do have very specific thoughts about how the scientific method can work with complex problems like the kind we have in the 21st century. But yeah, going for the ground truth, the fundamentals, being very systematic about things, being descriptive and really knowing what's going on rather than guessing. Yeah, I think there's a very specific scientific method that can be applied to complex 21st century problems. I guess at the middle of the high school age, learning the fundamentals is really important. Learn things that are still going to be true in 20 years. So I trained for 2 years in chemistry as a senior high school student / early college student, and then ended up majoring in physics and computer science. So I learned a lot of the fundamental sciences, which you can then use to solve problems. So that's a start, I guess.
That is a good start. And that's something that, I mean, anyone can do at any stage of their educational journey. For me, it was reading endless books like the ones behind me here, because science is fascinating. And if you just start with stuff that's accessible and easy and you find what you're truly passionate about, you can build up to having knowledge like Ed has and dive in and become a researcher yourself. So I can't encourage that for our kids enough. It's something that we hear a lot in our Gairdner series. So I do encourage you to check out Gairdner.org and add Gairdner Awards as well. And if you want to learn more about Ed's work, see those articles and more, synthneuro.org. Ed, thank you so much for your time, expertise, as always today. We really appreciate you taking any time out of your crazy busy days to hang out with us and share your knowledge with kids. And we'll just say thank you so much. And for our kids at home, have a wonderful rest of your day. We'll wrap up from there. Thanks, guys.